Welcome to Lab Presentation 1. Before beginning, you should have, first of all, watched the kidney model presentation. And if you haven't done that, you can go to Blackboard, go to Lab Materials, and go to A&P2 Models, links to videos. And before beginning, you should have also filled in your lab manual figures 48.1, 48.2, and 48.4, and looked at figures 48.5 and 48.7. So let's begin with figure 48.1 on page 374 in your lab manual. Take just a minute, first of all, and notice the renal arteries labeled for you and one going to each kidney. Find also then, and also labeled for you, the renal veins and one coming away from each kidney. Take a look then at the kidneys, number 1 on your model. Notice that the kidneys empty into the ureters, number 2, which empty in turn into the urinary bladder, number 3, which empties into the urethra, number 4. Let's take a look now at figure 48.2, also on page 374. And first of all then, locate the renal capsule, which is labeled number 8. And you can see then this is the surface of the kidney. Next, number 10, locate the renal cortex. And this is the layer of the kidney just deep to the capsule. And then locate number 9, the renal medulla, which in turn is deep to the renal cortex. And notice then that the renal medulla is composed of number 8, a series of structures called renal pyramids. Take a look next then at number 7, and we have these structures called renal columns. And you'll see that each column is an extension of the renal cortex located between two renal pyramids. Notice labeled for you now is the renal sinus. And you'll see this is a space enclosed by part of the kidney. Find number 5, the ureter. And then follow this tube, follow the ureter on up to number 3, the renal pelvis. And the renal pelvis then is the upper part, the upper portion of the ureter. Take a look next now at number 2, one of the major calyces. And notice each major calyx leads into the renal pelvis. Locate number one of the minor calyces, and you'll see that each minor calyx leads into a major calyx. And then find number 4, a renal papilla, and notice that each papilla leads into a minor calyx. Let's take a look next now at figure 48.4 on page 376. And we're going to first of all follow blood flow. So notice labeled in the upper right hand portion of the figure, we have a cortical radiate artery, and the artery is going to be the structure in red that you're seeing. Next, find number 2, an afferent arteriole. And notice then that this afferent arteriole leads into number 2, a glomerulus. And each glomerulus is a capillary system. And so you can see the branches, you can see the loops that collectively make up the glomerulus. Take a look next, and coming away from the glomerulus, you'll see labeled for you an efferent arteriole. And notice then that these efferent arterioles lead into, also labeled for you, a peritubular capillary system. If you follow that peritubular capillary system down, notice on the left we're seeing the portion of the system which is shown in red, and then notice on the right we're seeing the portion of the peritubular capillary system that's shown in blue. And this change then from red to blue indicates gas exchange. And so we're, as blood flows through the peritubular capillary system, gas is, oxygen is being lost, and carbon dioxide is being taken in. And then finally notice now the end of the right-hand blue portion of the peritubular capillary system in turn is going to lead into a cortical radiate vein. So again, that's blood flow. Same figure now, 48.4, and now we're going to follow the flow of a material called glomerular filtrate, and we'll follow the flow of that filtrate as it's converted into urine. So first of all, locate the glomerulus, number 2 again, and notice that the glomerulus is located within a structure, number 4, and this structure then is the glomerular capsule, also called Bowman's capsule. What's going on here, blood plasma is being filtered from the glomerulus into the capsule. And once this has happened, once the blood has moved out of the glomerulus and into the surrounding capsule, the blood, the plasma, which has been filtered out of the glomerulus, is now referred to as glomerular filtrate. And again, we're going to trace the flow of the glomerular filtrate. So notice, number one, leading away from the capsule is a proximal tubule, and you can see it takes lots of twists and turns. It's sometimes called a proximal convoluted tubule because of all those twists and turns. But follow now that proximal tubule, and you'll see that it leads in turn into a nephron loop, also called a loop of Henle. And notice then that the filtrate flows from the proximal tubule down the descending limb of the loop and then up the ascending limb. And from there, the filtrate, which is being converted into urine again as it takes this pathway, but the filtrate, the forming urine, next enters the distal tubule, and the distal tubule, in turn, if you follow that along, leads into a structure called a collecting duct, number 6. Finally, let's take a look at the figures that are micrographs. So page 377, figure 48.5, and notice now the A part up at the top, that photograph, we're looking at a photograph of the renal cortex, and you can see that within the renal cortex, this is where we find glomeruli. Looking now at the B photograph, now notice we're down into the renal medulla, and you can see here the absence of glomeruli and the presence of collecting ducts. And finally, let's flip over to page 378, figure 48.7, and you'll see this is a portion of the urinary bladder. Take a minute and locate the lumen, the space within the bladder, the space where the urine would be found. And notice then the labeling here is telling you that this lumen is adjacent to a mucus coat consisting of transitional epithelium. Deep to the mucus coat, there's a submucous coat, and then deep to that, a muscular coat comprised of smooth muscle. And notice also then we name this particular structure, this smooth muscle that makes up the wall of the urinary bladder, we name this layer of muscle the detrusor muscle. And that concludes lab presentation 1.